in which we have Dr. Aman Sharma with us. And he is the founder and CEO of ExoCan Healthcare, which is one of the most unique companies that I've come across, which focus on exosome healthcare based strategies. And it's a startup in India. And Dr. Sharma has various patented portfolios of EV techs and is in the and has been working in the EV community for many years now. And today he's here to talk about his journey as an EV entrepreneur and his journey from academia to industry, which is a very coveted area that we all need to, hear more about from. And today we have Emily joining in from Liverpool as a moderator, and I'm Swagatama joining in from India. So yeah, over to you, Dr. Sharma.
Okay, thanks a lot for this invitation, and I'm always and anytime happy to interact with the young audience, especially grad students, because being myself one few years back, I have understood and realized that how important it is to get a good mentor and good information at right point of time, because that makes a lot of sense in your PhD. It actually improves a lot of your research skills, as well as it gives you a lot of time to understand the concepts and the field as well. So good evening, good morning, and good afternoon, wherever you're based at. So I'm Aman Sharma. I am founder of this company, ExoCan Healthcare Limited, and we started this company in 2015, and almost now I think 7 years. We have been in this particular space. We are probably India's, not probably actually, in fact, we are India's first exosome company, and I actually wrote a first TV grant while I was in my PhD. So why not I start with one of the observation, and that will make some more sense. How I actually ended up in this field and not going for a postdoc instead. So if you see this slide, can you see the slide? Okay, wonderful. So if you see this particular slide, so this image was taken almost, I think, a decade back when I was mid of my PhD, and this is a co-culture of 3D cancer stem cells and endothelial cells. So while I was doing a PhD on brain tumor and trying to identify how brain tumor stem cells actually contribute to the aggressiveness of glioblastoma. So we made a co-culture system, and this is sort of we file a patent, and we developed a parallel technology around it. So we wanted to understand how the cells interact. So what we did actually, we labeled these cells, patient-derived cells, and with different non-genetic reporters, and we assembled a co-culture system which is self-replicating and not in a specific matrix. So it's like a suspension culture. So I did a lot of hacks to understand, how these populations interact and what kind of signaling is activated, which imparts, cancer stem cell resistance to therapy. But this is on the right side. If you see here, this is a flow cytometry plot, and if you see this particular quadrant Q2, you see those who are familiar with flow cytometry, they will appreciate the double positive population here. So this double positive population means, these cells are actually having both green and red color. Now, question, when I saw this plot first time, the question I was intrigued by the fact, how it is possible, because that time the exosomes and EV field was not, evolving at a pace that what it is right now. So there were very few reports and all these things. So we were having a seminar. A person from I think UCSD was visiting to give a seminar on exosomes, and around that time probably I got this data, and I analyzed this data, and I sorted this population, and checked in confocal and some other fluorescence technique also, and I found that indeed this population was actually having a dual color. So the question came to my mind how it is possible, and then as I mentioned, a bit of information on exosome was coming up that point of time. So I sort of purified this population, cultured, and did a bit of study on exosome isolation, and then characterization, and I found that this is indeed happening via exosomes. So this is the time when I started, this exosome word actually came into my mind, and I could actually foresee a career in this particular field. Now this was like mid of my PhD, and I had couple of more years, and I went to a conference in US, and I got a postdoc offer there. And when I came back to India, and I found that I had written a innovation based proposal, and I found that I got this innovation grant of approximately 60K USD. And with that, I actually started this company, ExoCan Healthcare Limited. And the questions that so can you see the upper heading, or if the pane is this? Can anyone comment how to, because this
It's visible.
It's visible.
Yeah, it's visible. Yeah.
Okay, so the existing method of EV or exosome. EV was not a very catchy word that time. Mostly people would use exosomes. Nowadays EV is more popular, so at places I would refer EV, and wherever required I will mention exosomes based on certain features. So these were the methods back then. Most of them were using, actually 70-80% of the people were using ultra, and the advantage of this method was that you will get a palette. Few of the groups were actually using in Europe. They were using ultra filtration based methods. The disadvantage with this particular method is, there is a very, people would argue back then that that lipoprotein particles will also come up with this particular filtrate if they are of similar size. And the major disadvantage was that it was a suspension sample. So whatever you get at the end is suspension. You can't, if you are doing a Western or you want to do some other kind of assays, you can't actually, if you resuspend again, you would dilute the sample. And so more feasible and user friendly was getting a palette actually. Then the third method people were using, most lab who were actually having decent funding, they were using precipitation quick methods. And the beauty of this method was that you could get actually a palette, and at the same time you could resuspend the palette as per your requirement. So part of it you could use for, let's say, electron microscopy or AFM or cryo-EM, or you could use for Western blotting, as per your requirement. So you could separate the palette in multiple parts and use as per your requirement. So now, when I understood, these are the existing methods and the challenges associated with these methods. So, when I started the company, the need was apparent actually. If you want to be an exosome company, you need to develop your own in-house method, which will be probably, I would say, user friendly, and it will not cost you more if you buy some external technology. So we wanted to develop a rapid, high yielding method which would capture the heterogeneity of exosomes and which is scalable. So if you can isolate a exosome from 2 ml or 5 ml, but what if you are unable to isolate 500 ml? Or the other way around, if you are able to isolate, let's say, using ultra from, let's say, 200 ml, but what if there is a sample volume very small, for example 200 microliter? So those were the challenges, and these were the questions we wanted to address. And the pitfalls, as I mentioned here, this time consuming ultra is very time consuming, and any graduate student or postdoc would avoid it, because until unless PA is actually forcing because of funding, so the yield is actually not too good. And there's a multi-step in case of ultrafiltration, then it would not capture heterogeneity. And in case of precipitation, so these things, most of the EV researchers who are probably listening now, they understand all these pitfalls. So now let me show you this particular picture actually. So this is ultracentrifuge tube, and you have a multiple. Now we appreciate recent 5 years, 3 years, probably there are reports that there is a lot of heterogeneity of EVs. From, let's say, EVs, when I say EV, like it could be 30 nanometer to 500 micron for nanometer, for example. And one method would capture one type of EV and another would capture another type of EV. So if you keep on increasing the speed, you get a different species of EV, from microvesicle to exosomes to exomers and supramers, and have you ever thought, after so much of spin, 40-50 hours of centrifugation, these EVs are feeling pretty bad actually. They are not happy. they are clumping and all things. So that with this increasing speed and time, you would also probably appreciate that there is a disturbance of protein lipid rough structures and protein, the bonds in the protein, and it might destabilize the proteins. And someday there will be a infinity K ultra and you get as transformers. So so this is all kind of merge you are going to get with the increasing speed of ultra when companies are actually making new machines. The takeaway of this whole couple of slides that I showed previously is that you need to have a good yield, you need to have a scalability, and you need to have a purity. If you are actually having this particular space of sort of Venn diagram, you are happy not as a EV researcher but as a EV enterprise also. Because if you have a yield, for example, if I'm going to detect a mutation which is pertinent to cancer, a specific cancer, and against which you have actionable drugs, you would always want higher yield of EVs or exosomes so that your mutation is actually detectable in your sample. But at the same time, you also want to make sure that there is a purity of sample. Otherwise, your signal will be masked by noise, which could be non-EV like proteinaceous particle in case of your biomarker is a protein. And of course, scalability. So I might develop a so I can feel the pain points of both of a researcher as well as a industry person, because I'm sort of standing in between. So even if there is a method but there is no scalability, hundreds and thousands of cancer patients are there. For example, take lung cancer. If your method is not, or technology is not scalable, you can't do ultra based diagnostic for, let's say, cancer, because it's not possible. So you need what? You need this kind of system. Basically, for example, these black circles, let's say these are X mutation carrying exosomes or EVs, and these are impurities, let's say. So you need to enrich first, which will give you or keep the information intact in terms of these mutation carrying EVs, remove a bit of uh, impurity, keep let's say and remaining impurity, let it be, and you have another method which can further purify without compromising on the information which is present in these mutation carrying EVs. And at the end, you can scale it up for hundreds and thousands of samples. So this is an idealistic EV or exosome pipeline, especially for example, diagnostic and for therapeutics as well. Now what we have got, and I'm going to show two of the projects that we finished and developed technologies around which, and which makes the basis of our whole startup. So we developed a 30-minute workflow which can enrich and purify EVs or exosomes, or I would say heterogeneous EVs at 5000 RPM. So you have a sample with different EV population in terms of size and information. You spin at 3000 RPM, 20 minutes, you get a palette for me. You resuspend as and when you require as per your requirement, and you further do a purification step of 5010 RPM 10 minutes as a benchtop centrifuge. What you get is an interface which will contain proteinaceous impurities, free nucleic acids, and the upper phase, aqueous phase, will have various size and cargo containing EVs. So this was our dream back then, 6 years, and the pipeline we completed a couple of years back pre-COVID time. So this gave us advantage and capabilities to perform a rapid EV enrichment from a variety of biofluids, a very high yield of 10 raised to 12 particles per ml, for example, in serum. These EVs are cargo enriched, a variety of RNA, DNA, proteins, lipids are present, and you capture the heterogeneity. It's a very critical part, because if we have this heterogeneity capture, we don't need to do rounds of ultra with different centrifugation speed to capture different sizes of EVs, and this will save a lot of time. And this is scalable. You can sample, do a 5000, even more sample. If you increase your capacity of lab, you can do more than 5000 samples a day. This part of this technology is patented in India, China, and US. So now how we developed, and what sort of takeaway from this whole exercise as a researcher come entrepreneur, freshly turned entrepreneur. So if you see this SEM image, so this image is a few years old. When we developed this technology, we found that from 68, 50, 83, 71, we could capture all sort of EVs in terms of heterogeneity of various sizes, and they were not clumped. So you remember, if you are having a soft landing, if you travel by a plane and you have a very hard landing, it's not pleasant, right? So we wanted to develop a method which will give a soft landing to EVs. So they are pooled pretty easily, and at the same time, they are able to we are able to capture their heterogeneity of various sizes in terms of whatever your sample is containing. So this is typical SEM image of heterogeneous EV sample. Then we extracted RNA out of it, and to our surprise, this is all sort of unpublished data, because as an enterprise we do not wish to publish but rather patent information. So if you see this 8 ml culture media only, and this right side is a 4 ml culture media representing 4 different cell lines. This is a silver staining of EV RNA. You see a lot of RNA, and I always wonder when in literature I find that the RNA is very less in EV. It's not the case. So of course we have done all sort of RNase treatment, post isolation, then purification. So it's ultra pure EV derived RNA. We also then subjected these EV samples to DNA, and we found that the different cell lines, and we found that this is a genomic. If you see this genomic DNA, and so the EVs indeed contain DNA which is like contrasting in nature. Some of the DNA will be small size, like probably this is a sort of unresolved 100 base pair ladder. So some of the DNA will be 100 to 200 base pair, and something would be bigger, like 2 kb to 5 kb and more. And these, when the report came in probably in 2016-17 from Raghu Kelluri group, where they showed that you have a large molecular weight, high molecular weight double strand DNA in EVs, we could actually see that yes, indeed, this our EV samples are actually containing both type of DNA molecules. And if you see this right side, so we modified the condition of the producing cell line. So let's say we have a EV producing cell line, and these are the 3 different cell lines. So we gave them different conditions, and we found them, when you gave different condition to your cell culture, the cargo export in terms of DNA in EV is pretty much different. So if you see this lane 1, there are 2 different unique bands appearing which are not here earlier. But when you change a condition here, so of course, because of some confidentiality reasons, I cannot speak what were those conditions. But it does happen that the cargo changes actually. And lately, subsequently, we found that lot of reports, they suggested that yes, the dynamic uploading or the dynamic loading of cargo in EVs. So then we isolated a protein, and there is this conventional method. So this is quite a 3-4 year old data actually. So this is, I'm going to tell how the whole story, how we actually develop all these things. So that time people were mostly using BCA, and then we did a BCA, we analyzed the EVs, and then we actually we could see the color is coming up, because in India back then there was no one actually, there were no reference lab, and even globally also, even I don't know now, but you would see that no paper published into till 2000, probably 15, 16, 17, and they would show a BCA wells, the protein wells actually, while estimating with BCA. So that, because seeing is believing. So once you see a color development, you see some, something is there in your EV sample. And then we did a blotting actually to see a specific this particular EGFR. We choose the cell lines which were highly expressing this marker, and we could see that it is preferentially transported, in this EV samples. This is a whole cell lysate actually. So then there was, even now it's sort of I would say controversial whether acetylcholine esterase is transported in EVs or not. But we developed this particular product, and we found that that the ACH is actually enriched in EV, even compared to cell lysate. So some of the data I'm showing here. So these are like just images. We are showing the rapid yellow color development, and this is a kinetic graph. So what the takeaway of this whole slide is that as a freshly started researcher come entrepreneur, we found the need evident, and we wanted to make sure we are independent in terms of our needs, and thus how we developed first product, which is ExoEnrich, which could capture EVs within 20 minutes at 3000 RPM without actually compromising on quality. Second product we developed for DNA isolation from EV, because we used various column-based products and we found that the DNA is lost from EV. Most of the DNA is lost. So, we wanted to make sure that we are capturing whole of DNA, whatever is there inside the EV. Of course, not outer DNA. So this led to development of second process know-how or a product for us. And we developed third product because, even now we know that EVs are pretty much resistant to detergent lysis. So if they are even stable at 1 pH of stomach, that's the basis of EV-based drug delivery. So they're pretty much hard because of their intact lipid raft structures, which is made by nature. So we developed this particular product which could efficiently lyse EV so that you get all of your protein, which is amicable for downstream proteomics studies. And the fourth product we developed for rapid quantification of EVs, at least in terms of how much is acetylcholine esterase is present in these EVs. So this particular exercise, within 1.5 years, we developed these 4 know-how processes or products, which actually help us establish further processes and technologies, few of which I'm going to talk in subsequent slides. So this is our whole portfolio as of now, a pretty competitive, pretty efficient across the globe, from isolation from various biofluids to analysis in terms of cargo, DNA, or quantification. Now we are a diagnostic company more, and a part of us we also do therapeutic EV based engineering. So as I mentioned, so if you have a EV sample, a typical lab would do isolate EV, then do a DNA treatment, probably RNA treatment, and proteinase K or whatever. So to purify further to see what is inside the EV, to remove outer contaminants. So no matter whether you're using ultrafiltration, ultracentrifugation, AFO4, or TFF, there are some impurities going to be there in terms of nucleic acid or some other biomarker molecules. So you have to go through typical standardization process, or you have to set up further reactions to get rid of those impurities. And that's a cumbersome process. It's not user friendly, and also lab scale it is good, but when you're going to develop a diagnostic, it is not going to help you. It is going to increase your processing time and remove the unique features or advantages of EV based diagnostic. So typically people also use ultra, which is the sucrose gradient 16-hour process, and not every lab would have it, and there's a long queue also, as I faced earlier in my graduate time. So there is a scalability issue indeed, even if you are developing engineered exosomes for therapeutics or diagnostics. What we developed a 10-minute process actually to isolate, purify EV for biomarker discovery and diagnosis. So you have a EV sample, and you just use a simple process of 10 minutes, 5000 RPM. What it will do is will separate the phases. Upper phase will have your purified EVs, as I mentioned in previous slides, and in between you will have sort of impurities, and you take up the upper aqueous phase and do a lot of characterizations to check if your EVs are intact, they are functionally valid, and they are containing unique biological cargo useful for diagnostic purpose. And this process is scalable from 1 ml to 1 liter, and you could do even more. This is a typical image. If you see here, we have used different concentrations of exosomes or EVs. So this, you see the interface here, typical we see in case of DNA or some other kind of purification methods. So this is the interface here, if you see, and this is organic layer comprised of protein-free nucleic acids, and upper phase you will have purified EVs or exosomes. So what we did actually to understand how much BSA, because mostly if you are using a serum-based cell culture, the most proteinaceous contaminant is BSA. So what we did initially, we used a buffer and spiked with BSA and use our system and separated the phases, and we took the interface plus the upper phase and calculated how much BSA we can precipitate from the sample. So same thing we did for EV sample also, and we found that if your concentration of BSA in the sample is up to 5 microgram per ml, which is a typical case, let's say in any exosome sample, even it's a pretty high 5 microgram, you won't even get a 5 microgram per ml protein in EV sample. But let us assume this is the upper limit, you are able to get rid of 99.93% of BSA from your EV sample. And if you have more amount of BSA, so efficiency goes a little bit down, but still you're able to get rid of 90% above BSA. So then what we did actually, we took the EVs or exosome from upper phase and did SEM to check if actually they are present in the upper phase or not. We did NT as well. So if you see this electron micrograph, scanning electron micrograph, they are very well dispersed EVs of different sizes, smaller, bigger, and even bigger. So we are keeping the integrity of the integrity of and heterogeneity of EV sample intact while removing the proteinaceous contaminant from our sample. Then what we did actually, we loaded these various cell-derived purified and non-purified EVs on a SDSP gel and CBB stain to see BSA band. So if you see a 68 kDa band, so this is your purified sample, and this is non-purified samples of various cell lines. You see the BSA band is actually disappearing. That means we are actually able to get rid of BSA band from the impure EV population or sample. Then we did a lot of other markers also, 3 MISCV recommended markers. But I'm just representatively, I'm showing flotilin 2, which is the lipid rough marker for EVs. So if you see here, purified EVs versus control EVs, and then this is a whole cell lysate, and again for another cell line purified and control EVs. So most of the flotilin was intact in this particular sample, making sure that the membrane is not compromised. Then we isolated DNA from these EVs and did a UV visible spectrophotometer, and we could see the 260 nanometer typical peak of DNA in purified as well as control EVs. So we isolated the DNA and did a later on we isolated RNA as well and did a GAP-DH PCR to check the transcript level, and this typically may show that purified versus non-purified EV at least maintain the housekeeping genes. There is no compromise of membrane integrity. So this whole data shows that purified EVs are intact and cargo is intact inside the EVs. Then what we did, the interface I showed you, the interface we took, and we wanted to see what is there in the interface. So if you see the scanning electron micrographs here, a different concentration of exosomes and in the sample, you see that there is a proteinaceous crystals of mostly I would say protein nucleic acid aggregates in the scanning electron micrograph, and in the background you do not see any EVs or exosomes, ensuring that most of the population or exosomes are purified and impurities are trapped in the organic layer. So this gave us confidence that yes, the method works, it is scalable, and it can be used to purify EVs instead of sucrose gradient in a simple 10-minute process at a very low speed of 5000 RPM. Now, some of the data I'm going to show you, which we recently developed as drug loading technology. So as we all know, the basis of exosome-based, engineered exosome-based drug delivery is that, you have a cell of interest, you have a target receptor, and, there's a typical EV surface protein. You make a chimeric fusion protein, which is your upper side of, let's say, target ligand, which is cognate to this receptor. So this is a sort of surface display technology of exosome engineering. Most of the labs are using currently across the globe, and even companies also. And then you set up a batch production, you isolate these engineered exosomes, load your drug, further purify after loading, and use for therapeutics or in vitro in vivo study. The real problem comes here, so that there is no method globally available for drug loading. So what we have done in that particular segment or space, I'm going to show a few of the data here. So let's say you have engineered exosomes. Current methods, there are no scalable industrially useful methods so far, and lab-based methods are sonication, electroporation, and detergent-based methods. There are typical pitfalls: segregation, poor efficiency of drug loading, and you have a lot of membrane integrity loss of EVs, and making them non-functional for the downstream studies, and there is no batch consistency. So there is no scalability. We developed a two-step scalable process for cargo loading in EV, and one particular drug data I'm going to show. So this whole process takes 1 hour of time, and we just simply do a surface modification of EV to load this cargo. So we have already two technology established, as I talked in the previous slides: EV isolation and purification. So now with this EV cargo loading, drug loading, so before and after drug loading, you need to purify EV. So we had all these technologies in place. So with this technology, we could actually complete our workflow. So we wanted to see how much drug we are able to load. So this is a typical workflow of EV isolation garage. So most of the industrially useful and therapeutics EVs, people are using mesenchymal stem cell derived exosomes or EVs. So we isolated, grow. This is typical primary culture. We did a bead-based characterization of various couple of exosome markers, and did a blotting also to check the purity, and did NTA, like we got a band of 80, 70, 980 nanometer major peak. And so this is a so once we isolated these EVs from MSC, now we wanted to load drugs. So I'm going to show in subsequent slide how we achieved high drug loading without aggregation and retained functionality in batches. So we use sonication, electroporation, and ExoCan, our approach of drug loading. And you see this. So with our technology, we get a visible palette of EVs. So you see the visible palette here. In 3 methods, we got a visible palette. But what we got very nicely is that in our approach, we see almost 47% of drug loading of cisplatin in these EVs, as compared to sonication and electroporation, which are between 12 to 18%. This is a almost threefold, two to threefold increase in drug loading, and industrially useful for us. Now we have loaded the drug now, so I'm going to show a few of the data now. So definitely, because of confidentiality reasons and all, I'm not going to show whole of the data. What I'm going to show is representative data. So once you load the drug, you need to make sure that the shape and structure is intact, okay, and your membrane intact of your EVs, so that there is no drug leaching. Your drug is intact in the EV, and your batch is stable, which you get by a simple in vitro IC50 determination. Let us see how it works. If you see before loading and after loading, we get a very nicely dispersed SEM image of EVs. There is no aggregation whatsoever after loading, and this is before loading. So we wanted to check the membrane integrity. So this is a lipid raft flotilin 2. So you see there is no decrease whatsoever in the pre and post drug loading. That means membrane was not compromised. And this is EGFR is a lipid raft, also transmembrane receptor. So if there is a membrane compromised, so you might lose this particular receptor in terms of quantity. So we don't see any great difference in the two bands, pre and post drug loading. So this ensured that IVs are pretty much stable and non-compromised at the membrane level. And we also did NT. We get a 17 9 nanometer major peak, as I shown in SEM also here. There is no aggregation, so we get a very well contrast peak of post drug loading of EVs. So now we wanted to see the batch stability of these EVs. So we stored these EVs for various days, and we did a simple in vitro assay to see if after storage the drug in the EV is actually similarly efficient, or if the activity of drug is decreased. So we did a simple IC50 analysis, and if you see here, there is not much of IC50 difference from 11 to 17 if you keep for 5 days. And mind it that it is not with any EV protectant. It's just pure drug loaded EVs we stored without using any sucrose or trehalose. So it's just pure. So if you have some additives, you can actually increase the stability of this batch further. So that will help you actually keep this IC50 more stable. So even now it is not much of change. So then we actually made different batches, like one batch, another batch, and another batch, and stored for different days, and we compared across batches. And we by measuring the content of the drug in EVs, free drug versus stored EVs. So if you see here, even increase the storage time 1 to 5 days, different batches across different days will not have much of difference. That means almost 90% of your drug is stable inside EVs. So now if the drug is functional or not, so cisplatin is a DNA damaging drug. So we wanted to see if you incubate these EVs on cells, are they going to induce drug DNA damage? So if you see the staining here for gamma H2X, so if you see gamma H2X foci, they are in cisplatin control drug or EV loaded or exoplatin drug. So both are actually able to make gamma H2X foci. So that means drug is actually intact, EVs are functional, as well as drug is functional. And if you also see the flow cytometry data here, so G0 G1 arrest was not changed much. So from 60 control 64 to 61. So that means if your drug is stable, you won't see any kind of change in the arrest, cell cycle arrest. So this actually gave us confidence that yes, this technology works and it can be scalable, as well as it is keeping the drug intact inside the intact PV post drug loading. So I think this is a few of the non-confidential work that I could share, and I would like to thank BIRAC Government of India for funding support and NCL Innovation Park for providing us incubation facilities. And I would be more than happy to take more and more questions from you people, because I prefer I speak less and you people speak more, because you are working day to day with the EVs. So I'm happy to take question, and thanks a lot. Thanks all for your attention.
Thank you so much, Dr. Sharma. This was a very amazing talk. Before we go into the Q&A session, I just want to start off with my one question first. Apart from the, from the need of wanting EV technologies in India, was there any other reason why you shifted to industry and take up academia? Because we all know the usual hurdles that there is in industry, but then you went ahead with entrepreneurship altogether. So how was the overall climate at the time when you started this organization? What all hardships did you face initially?
So you are let's say you are asking the challenges associated with
Yeah,
setting up
with
an
opening up a
enterprise.
biotech. Yeah.
So it's not easy to deny a Harvard postdoc offer that is coming from one of the top 2% lab in brain tumor research, and probably a UCSD offer where people are publishing in Cell Nature quite often. The only reason was, because I had no choice. I was very clear from the very beginning of my PhD that I wanted to do translational work, and but of course you needed a support and funding. So it is ironical that I submitted thesis, let's say in May, and I had a my lab already functional in April. So before I would submit my thesis, my lab was functional here. So because of the funding that I got, and I was always ready to take up challenges, and this field was pretty much new, not just in India, globally that time, 7-8 years back. And I could find a lot of opportunities and possibilities, provided we could solve some of the main hurdles in EV research, which I had shown in this presentation. So if you are ready to take a challenge, it won't matter whether so at first slide I showed just a facts co-culture experiment followed by flow cytometry. It could spark your idea, and if you're good at reading literature, I think you would anyway get idea whatever experiments you're doing day in and day out. And if you're ready to take up risks, it's a very it's a much rewarding career, of course.
Right. Yeah, we're looking forward to a lot of challenging hurdles that come up in the future for each and every one of us, and I hope we deal with each and every one of them with the same enthusiasm. So I think we can pick up the questions. We have a couple of questions, I think. Yeah.
Brilliant. Shall I take the questions?
Yeah, if anyone wants to ask that question, that's fine as well. If you want to just have a discussion with Dr. Sharma, that's perfectly okay. But I think we can just read them out for now.
Definitely. Whilst people are deciding, I have a slight question. You mentioned how obviously going into sort of the industrial side of things rather than academia, you the switch between publishing papers and instead you want patents. And I thought that was really interesting from I don't know if anyone else feels like this when they're in academia, but obviously there's this huge push to publish. And I just wondered if that was like a very big shift for you in terms of what your research priority was in terms of the output and where it goes.
It was not a big challenge, as I said, and it was not there was no two options for me, because as I mentioned, if you have ideas and you get a funding at right point of time, the transition would not be difficult. The only thing is difficult how you set up your lab. So even your postdoc after 10 years would find it difficult to establish a lab. Those skills you don't learn in your graduate studies, or probably even in postdoc, that how to order machines, develop SOPs, develop technologies, write patents. These things across the globe, no academic person, be it grad student or postdoc, will learn. So such things comes naturally, innately, with your inquisitive nature of making research from bench to bedside. So and as and when you progress in your career, you learn these things. So uh, I don't feel like I faced much of challenges while
Mhm.
having this transition. The only challenge was how to secure the next funding. That's all.
Yeah, that makes a lot of sense. Would those that have put questions in the chat like to speak, or we'll take, if you don't unmute yourself, that you'd like us to read them?
Sorry, are you asking me?
Hello, I'm Sangati. Hi, it was a very nice talk, Dr. Sharma. I hope my voice is audible.
Yes, yes.
Yeah, so I had a question regarding the EV yield that you mentioned in one of your slides that, you collected the 4 ml and 8 ml of the conditioned media, and then you went ahead with the Western blot. So I had a question that what was your preliminary EV yield from, this much of conditioned media? Since you have mentioned already that EV yield, purity, and scalability was then, more in your case. So since our lab is facing the some kind of challenges with these right now, current ongoing our PhD studies. So I would like to know more on this so that it can be helpful to our studies too. So that is one thing, and second thing I'll post later on.
So yield is critical, as I mentioned, any kind of assay. So that time, people were reporting 10 raised to 7 particles and all. So there is a lot of scale issues also. When they say per ml, they don't say per ml from media or per ml of suspension of EV from X ml of media they got. So the per ml concept is pretty much confusing in literature even now. So we wanted to rationalize, and we found that from let's say for 2 ml of culture media, you could get more than 10 raised to 12 particles. So and so they will be of different sizes, and they'll contain different sort of cargo. So and if you let's say purify and do a protein estimation in BCA, you would get a very high yield of protein, up to 50 microgram and more. So uh, one of the thing that actually helped us establish different technologies and all, because of simple fact that we get a very high yield in terms of particle number, in terms of protein, and in terms of cargo that it contains, be it RNA or DNA. So I always wonder, people report they get a nanogram of RNA and EVs and all, and we get almost close to microgram if we use, let's say, 4 ml of culture conditioned media. And of course, after doing RNA treatment and all and purification, it's not just it's only outer, not the outer RNA. It's of course internal RNA. Yeah.
And that too from 2 and 4 ml of conditioned media after centrifuging it twice or thrice, whatever it is that you mentioned. So this was the yield up from this itself 2 and 4 ml of media.
Yes, yes.
Oh, and good enough to go ahead with the Western and NTA characterization. So we'll definitely look into, because I think we have been facing issues with our yield also. I may have missed out on one of your slides that you previously you had carried out your studies of EV isolation through ultracentrifuge purification method.
No, we
Or
did, once we were when we were establishing the technology, we had to compare with the gold standard, which was ultra back then. So
okay.
the ultra yield was definitely, the scale is like, one is like, for example, the scale of, if you compare two scales, one is having a unit 1 and another is having unit 100 or 1000. So we had such a scale disparity, like the ultra was giving very poor yield, so compared to our technology.
Okay. And just one last question from my end. Since you had also mentioned that there are various other ways through which we can isolate our EVs. So one such way is through many commercial kits that are available. So one such thing that I had in my mind, that how is ExoCan kit that is your product is different from the commercial kits which are based on the precipitation method. So wanted to know a little bit or elaborate more on, could you please elaborate more on this aspect, that how is it different? Since I know that, it gets separated into phases, but just a little bit more elaboration on, I may have missed out some part of it in your presentation.
So
Thank you so
phase separation is a purification step after isolation. And the isolation, your question is more pertinent to isolation methodology. So we disturb the ionic strength of the media or sample, followed by a simple cooling down at a very low speed of 3000 RPM. So, it is like a soft landing. We disturb the ionic strength, the structure is disturbed in the solvent system, and then we add second reagent to sort of precipitate or cool down the EVs at a very low speed.
Okay. Thank you so much.
I see we've got one more question in the chat. I'm not sure if that individual would like to say the question, or I'm more than happy to. It's entirely up to you if you want to unmute or not. I'm quite happy to go for it.
Yeah, so we are working mainly based on proteomics, especially from plasma samples. And one of the challenges that we are facing is that even after density gradient, we are not getting pure EVs. And since we are working on biomarker, we would like to get rid of the contamination that are especially coming from plasma. For example, the complement proteins, the coagulation proteins like fibrinogen and apolipoproteins. So I was just wondering whether you have tried this kit on the plasma samples and whether you have any proteomics data suggesting that your product is better than the ever evolving isolation techniques, and whether it is actually able to get rid of all the plasma proteins that are not EV associated.
So, so first of all, there is no method in the world which is 100% accurate, right? So I would not say all the plasma protein, but we have been doing this work on serum samples also. So, and since we wanted to build a Indian EV community, so with that motto we started disseminating these product range to Indian EV community, and you can write me and I can show you the references. People have published in high impact journals where they did proteomics, transcriptomics on serum, cell culture and whatnot. So that data is already in public domain.
Okay, thank you.
Brilliant. So does anyone else have any questions? No, that's fine as well. Do you have any more questions, Swagatama?
Not so much as of now. I mean, I think this entire thing is sort of a new area for me to, understand, because the way in which the entire optimization has happened, if we get to actually use that in our lab, it would be fantastic, because we usually lose a lot of time and the scalability is a major issue that we're facing. So this is definitely, a step in the right direction that I think I'd like to work on in the future. So yeah, I think that's about it. And if there are no more questions, we can maybe ask all the audience members if they want to put their video on, and if we can take a quick screenshot. So we just need to take a picture and post that on the social media. So if that's possible.
Camera is actually some creating problem, I think. So that's why I turned it.
Okay. Okay.
Let me try to stabilize it. If give me a moment, I'll just.
Yeah, sure.
Yes, you can post.
A lot of us are joining from the labs. I can see that. So that's great. All right. So I'm going to take a screenshot and then we can just move on. Okay. Great. Got it.
Over?
Yeah.
Okay.
It was really a pleasure having you speak about the entire aspect of EV, EV field in India, and having us represent the work as well. It was really glad to have you with us, and we hope you get to attend the future SNEV events as well, Dr. Sharma. I'm really glad that our team could reach out and work this out for you as well as all the other participants. So thank you on behalf of the entire SNEV community.
Thanks a lot. It was indeed wonderful interacting all of you.
Thank you. Thank you
so
so
much.
much.
Thanks a
Bye.
lot. Thanks. Bye.
Thank you. Bye-bye.
I think that went
I
well.
think went well, yeah. Are we still
Definitely.
recording?